Hey y'all, Dr. G here. We are now moving on to our fourth video in our series of nucleic acid videos. It now makes most sense to move on to some kind of structured slides because the images that we're going to look at are pretty hard to draw by hand. So up to this point, we focused in on the general structure of nucleic acids, focusing in on the themes that we see, the phosphate backbone, the ribose sugars or deoxyribose, the presence of the bases, the structures of the bases, and how the polymerization reaction happens. In this video, we're going to focus in on the structure of DNA and RNA polymers. Okay, so it's probably no surprise to you that they end up forming into the very well-known and famous DNA double helix. Okay, so what I'm pointing out here is some important aspects of this. So this image points out the sugar phosphate backbone, right? So this is a ribbon diagram, just like we saw with proteins. We can trace the backbone, in this case, the phosphodiester backbone, shown as a ribbon. Okay, notice that it's an opposing polarity. So these two chains come together. One of them goes 5' to 3'. The other one goes 3' to 5'. So they have opposing polarity. Okay? They make the well-known hydrogen bonds. So G and C base pairs making 3 hydrogen bonds, A and T base pair making 2 hydrogen bonds. And those hydrogen bonds we talked about in the second video when we looked at the structure of these nucleic acid bases. Okay? Other important things to recognize here in this structure is that we end up forming some very well-defined grooves. We call it the major groove and the minor groove. And when I show you the actual three-dimensional representation of the DNA double helix, it'll be very clear to you why they're called the major groove and the minor groove. The major groove is wide and deep, and the minor groove is much more shallow. Okay? And the last thing that we'll talk about that really matters here is the idea that these base pairs that happen with our nucleic acid bases, they stack together, as you can see here, right? So this base pair is stacked below this one. There's a stack that happens all throughout. And as it turns out, this stacking interaction is by far the most important in stabilizing DNA, the energy that stabilizes DNA. So here we're looking more closely at our base pairs. On the left, we have an AT base pair. Okay, and notice here the thymine has this methyl group hanging off, which helps us know for sure that it's thymine, not uracil. And on the right side, we have a GC base pair. Okay, as expected, as we talked about in our nucleic acid bases video, the AT makes 2 base pairs, as we can see here, and our GC makes 3 base pairs. Okay? Let's look and make sure that these structures match our predictions that we made earlier on. Remember adenosine? I said it's a donor on top and then an acceptor. So here, this NH2 group is donating the hydrogen bond we see here, and this nitrogen is accepting the hydrogen bond. So with adenosine, we have a donor-acceptor pair. That's exactly what we expect based on what we talked about in the themes of these things. And the guanine, we have an acceptor, a donor, and a donor. So this oxygen is accepting the hydrogen bond. This is donating the hydrogen bond. This is donating our hydrogen bond. Okay? Another really useful, very useful thing that comes out of this is this idea of Chargaff's rule. Okay? Since is always ATGC, always one purine and one pyrimidine in a base pair. The ratio of purines to pyrimidines always must be exactly 1. Okay, that is 100% true in chromosomal DNA. Whenever you have a DNA double helix that adopts this canonical structure, it has to be made up of a purine-pyrimidine pair. So the ratio of these is always exactly 1. So here we're seeing our ribbon again, right? So you can clearly see the ribbon that represents the sugar phosphate backbone. And what this image is focusing in on is the idea of the pi stacking here. So I'm doing this is representing a space-fill representation of the nucleic acid bases. You see they're all planar because everything is hybridized sp2, which is a planar hybridization. So these things stack up really, really nicely. And if we rotate it and look and actually look at it from the end on, so looking at it down this axis, we see here that it's very clearly stacking on top of each other, and there's no space down the middle. If we rotate it and look at it from the side on with the same space fill representation, we see very clearly the two grooves we were talking about. The major groove is wide, right? It's quite wide here, and deep. The minor groove is much more narrow, and it's not as deep. Okay, so these are some really dominant characteristics that we can observe when we're looking at the structures of these nucleic acid polymers. Okay, what we just looked at was called B-form DNA. Okay, and it's the most dominant form. It's the only form that exists in DNA double helices. Okay, that's represented here, the B-form DNA. We see our major groove, we see our minor groove. This is what we expect when we think about DNA double helixes. But as it turns out, it's not the only form that exists. Other really important forms exist, most notably what's called A form. And A form is the shape of the helix when just RNA forms a double helix or DNA RNA hybrids. So A form is the most dominant form for RNA double helices or DNA RNA hybrids. Okay, and structurally, you can see some pretty major differences, right? So it may be hard to see here, but this A form helix is wider. It's fatter and it's compressed relative to the B form DNA, which impacts the major groove and the minor groove structure. The minor groove in A form DNA is almost non-existent, and the major groove is much skinnier. Okay, and it's not quite as deep. All of this happens because of something known as sugar pucker. So if we focus in on this, on the table, and really think critically about sugar pucker and distance between base pairs. In B form DNA, the deoxyribose adopts a different sugar pucker than in A form. So in B form DNA, we see here that our 3' hydroxyl group is kind of oriented out and the 2' is up. If we're dealing with A form DNA, then as it turns out, this 2' OH group is oriented down and the 3' is oriented up. So the big take-home message is that the sugar structure is different, and that impacts the distance between these bases. So in A form DNA, the bases are closer together than they are in B form DNA. For that reason, the A form helix is compressed. It widens, it fattens out, and then it gets compressed. So if we look at the structure of the A form helix, we can see some very dominant differences. I'm going to skip this one and go right here. Most notably, the major groove and minor groove looked very different. Okay, and even if you can't spot that right away, if you go back and look at this image for B form DNA, it's quite different. We also see that because this helix is kind of swollen and compressed, we actually get some space right in the middle that we can't actually fit anything down there, but there is space, which is very representative of the idea that this helix has swollen. Okay, one last thing that I'll leave you with with this video is that DNA structure doesn't just stop at the double helix. There's other things that happen. In prokaryotes, the DNA compresses further using something known as supercoiling. So the idea is represented by this image pretty well. So if we have a circular chromosome, right? The idea is that this doesn't float around as a circle. It coils down, compacts on itself, and it gets supercoiled. This serves a few purposes, one of which it's much more compact and doesn't take up as much space. Another purpose is that it prevents degradation, right? So because it's compacted, not as much surface area is shown, is exposed, so it can't be degraded as easily by enzymes or other things that are trying to degrade it. Okay, in the last video in this series, we'll focus in on the structure of DNA in eukaryotes and see how it's a little bit different.